All right, let's dive into one of the most incredible stories in all of biology, the first week of human life. We're going to trace this amazing journey, starting from a single cell, the zygote, all the way to a complex little structure that's ready to make a home in the uterus. It is a mind-blowing transformation, and it all happens in just about 7 days.
So take a look at this. This map lays out our whole journey. It all kicks off way up in the oviduct with fertilization. From there, this brand new embryo starts traveling down the fallopian tube. And over the next week, it's dividing, changing, and reorganizing itself until it gets to the uterus as this thing called a blastocyst, which is just about ready to hatch and implant. Let's break down exactly how it pulls this off. So, here's our game plan. We've got 6 key stages to walk through. First up, we'll look at the initial cell divisions. Then we get to the really cool part, when the embryo's own genes wake up. After that, we'll see how the cells start talking and working together, how they form that first big structure, the blastocyst, and how it gets ready for implantation. And finally, we'll peek under the hood at the metabolic engine that powers this whole trip.
Okay, so our story really begins at that moment of fusion. When the sperm and egg's genetic material combines, you get a single diploid zygote. Diploid just means it has the full, complete set of chromosomes. And this one cell, believe it or not, gets to work almost immediately, starting a series of super-fast cell divisions called cleavage.
Now, these first few divisions create the first embryonic cells, and we call them blastomeres. But here's the wild part. The embryo divides from 1 cell into 2, then 2 into 4, and so on. But the whole thing doesn't get any bigger. It's all happening inside this little protective shell called the zona pellucida. So you get more and more cells, but no actual growth in size. It's all division, division, division.
For the first couple of days, you can think of the embryo as running on a pre-loaded program. It's using all the genetic instructions and materials that the mother packed into the egg. But that can only get it so far. To really get development going, it has to fire up its own engine and take the wheel.
And that critical moment is called zygotic genome activation, or ZGA for short. This is where the embryo essentially boots up its own operating system. It clears out all those maternal instructions and starts reading its own unique DNA for the very first time. This is the moment it truly becomes a genetically distinct individual, driving its own development.
So let's look at how this all plays out over time. Right after fertilization, you see a little bit of gene activation. But the main event, the major ZGA, happens in us humans right around the 72-hour mark. The embryo is only about 4 to 8 cells at this point. And once that genetic switch is flipped, it paves the way for the next major physical changes, compaction, and then forming the blastocyst.
What's really interesting, though, is that this timing is not the same for everyone. ZGA is fundamental for all mammals, for sure, but when it happens varies a lot. In mice, it's super early, at the 2-cell stage. For humans and cows, it's that 4 to 8 cell window. But for sheep, it's even later. It just goes to show you how different the developmental clocks can be across species.
Okay, so the embryo has its own instruction manual running now. What's next? Well, it's time for its first big construction project. It's about to go from being just a loose clump of cells to a tightly knit, communicating unit.
This process is called compaction, and it's exactly what it sounds like. The cells literally change their shape. They flatten out against each other, and suddenly you can't really see the individual outlines anymore. They're not just a loose bag of marbles anymore. They're becoming a single, solid team called a morula.
And 2 things make this possible: intercellular junctions. First, you have tight junctions, which are like superglue. They seal the outer cells together, creating a barrier. But just as important, you have gap junctions. Think of these as direct communication lines, or little tunnels, that let the cells pass signals and molecules back and forth. This is how they start to coordinate their actions.
So how do we know these communication lines open up? Well, check out this experiment. This is the before picture. A researcher injects a bright yellow dye into 1 cell. See how in these early stages the dye just stays put? It doesn't go anywhere. That tells us the cells are functionally isolated. The phone lines aren't connected yet.
But now look at this. This is the after picture. At the later blastocyst stage, you inject that same dye into 1 cell, and whoosh, it spreads like wildfire to all its neighbors. This is beautiful visual proof that those gap junctions are open for business. The cells are now in constant communication, ready for the next big step.
This new ability to talk to each other and stick together sets the stage for the embryo's first and maybe most important decision: differentiation.
This is where cells stop being jacks of all trades and commit to a specific career path. They're about to split into two distinct types, and in the process, they'll create a whole new structure.
This whole thing is called cavitation, and it's just an elegant piece of biology. First, those outer cells, which we now call the trophectoderm, use their tight junctions to form a perfect seal. Then, they start pumping sodium ions into the middle of the embryo. And what does water love to do? It follows salt. So water floods in, creating a fluid-filled cavity called the blastocele.
And what we're left with is the blastocyst, which has two very different groups of cells. Tucked away on the inside is the inner cell mass, or ICM. These cells are pluripotent, and they're going to become the embryo itself, the actual baby. The outer layer is the trophectoderm. Their job is to be the support crew. They'll form the placenta and all the other tissues needed to support the pregnancy. This is the first great fork in the developmental road.
So now we have our blastocyst. It's beautiful, it's organized, but it's still trapped inside that protective shell, the zona pellucida. To have any chance of implanting in the uterus, it's got to break out.
And this is how it happens. Just look at this incredible image. This is a real human blastocyst literally hatching. It does this by pumping in even more fluid, which makes it expand and creates a ton of internal pressure. At the same time, it releases enzymes to weaken the shell. Eventually, pop, it breaks free and is finally ready and able to implant.
Now, a quick but really important detour. In science, we often use mice as a model for human development, but it's crucial to remember they are not the same. Just look at some of these key differences. The timing of ZGA is different, implantation is different, and maybe most importantly, the whole shape of the embryo after implantation is completely different. We form a flat disc while they form a cup-like cylinder.
And you can really see that difference here. On the left, that's the human setup. Our trophectoderm forms this really invasive tissue that literally digs into the uterine wall. But on the right, you have the mouse, which forms these giant cells and a totally different kind of placenta. These aren't just minor tweaks, they're fundamental differences in strategy.
Okay, so while all these amazing physical changes are happening, there's a whole other drama playing out in the background. It's a complex, delicate biochemical dance that's fueling every single step, and it is incredibly sensitive to the world around it.
This diagram shows just how tightly metabolism and genetics are connected. The embryo takes an amino acid, methionine, and processes it through this pathway. One of the byproducts is used to attach these tiny chemical tags, called methyl groups, directly onto the DNA. These tags act like little sticky notes that tell genes to turn on or off. This is absolutely essential for things like genomic imprinting and making sure the right genes get activated at the right time.
This also shows just how clever the embryo is. See, when it uses amino acids for fuel, it creates a toxic waste product, ammonia. Inside the body, that's no big deal. It gets whisked away. But in a lab dish during IVF, it can build up and become poisonous. So the embryo has its own built-in recycling plant to detoxify the ammonia. It's a perfect illustration of why the exact chemical makeup of IVF culture media is so unbelievably important.
So when you step back, this first week is just a masterpiece of biological programming. It's a cascade of perfectly timed events, and while it's incredibly resilient, it's also exquisitely sensitive to its environment. As our ability to support this journey in the lab gets better and better, it really makes you think about the huge responsibility we have to get every single detail right, not just for a successful pregnancy, but for the lifelong health of the people who start their lives this way.